computer. It also seems like someone is writing on the screen. Well, welcome everyone to the first of our initiative, the KZN eStream Cyber Club. It's our first workshop with STEC at UKZN. And you all know, I'm sure most of you have attended a workshop with Dr. Tanya Reinhardt. So I'm just going to do the first part explaining the expo and how we fit in, and then Tanya will do the actual activity. Samkela, if you can help me with muting participants, please. Someone's microphone is okay. So basically, the expo, what is the expo? For those of you that don't know, if it's your first time, anyone first time attending a session of this sort? Anyone? You could just raise your hand or put a note in the chat. If this is your first time hearing about the expo, right? Just a quick one. The expo is a science fair for learners, an opportunity for learners to present their own scientific investigations or engineering projects. This is, you can see learners presenting their work. This learner participated in Taiwan, and she actually came back with a third place award. For some reason, this thing is stuck. Well, this is last year. This is one of our regional expos in KZN. This was the one in, I think it was Newcastle. This is the one that we had in Durban. As you can see, this is the layout of the expo. And this is the competition day of the expo itself. But prior to that, there's a lot that needs to be done. Why should you participate in the expo? There's a whole host of skills. I'm not going to go through all of it, but just to tell you, it's an amazing opportunity for learners. Whether learners win anything or not, they will get a certificate, but it is a process of developmental. We've seen learners that participate in expo, we've seen their academic results improve. And they excel when they go to university because they're doing these research investigations by themselves. That's the key with the expo, is learners get the opportunity to be the scientist, the engineer, and the researcher while they're still at school level. So teachers, there is something in it for you as well. You can earn SACE points. And of course, there's opportunities of participating, representing the province, representing the country, and things like that, right? So this was last year's ISF. These were all the gold winners at ISF, which is our national and South Africa's international science fair. And there you can see our KZN learner Thea here. She won quite a lot of prizes. You can see she's standing there with some of her prizes. So I'm just going to run through this very quickly. This is one of our success stories. And I want you to go and read up. Those of you that don't know about CSO, go and read up about him. He actually has got a planetary body named after him because of his Expo project. And he says here, I'm an example of what happens when you give young South Africans opportunities. And this is what the Expo is all about. Giving opportunities to young people. And this is why we want to, this is basically why we're promoting the Expo. So very quickly for the grades that can participate, these are the grades. Learners can participate as individuals or pairs from grade 4 to 12. This is some of the written work that the learners need to have. And the first thing the learners need to start with is the research plan. Right, what is this eStream Cyber Club all about? So the idea came about when we looked at the categories for the expo. And we tried to make the Cyber Club, because it's online, to cover the various aspects that we found learners interested in. So lots of schools have the enviro clubs. They might have an art club or a maths club. So we tried to bring all these school clubs into the eStream, right? So I've just told you, what is the eStream Club about? We're going to be presenting online activities. Who can join? This eStream Club is specifically for school clubs. And the idea is to make the expo, which should be one of the activities, sustainable. So we want to do activities with the school clubs, and in that way, the learners take charge of the club and Expo becomes sustainable. Why should you join? Well, that's for you. You can decide whether you want to be part of these online activities. Okay, so school clubs, if you haven't started your own school club as yet, please do so. This is the guidebook. You can download it or you can email me and I can send you the handbook on how to start the science club. But in order to join the STEAM club activities, you need to have a school club. So that's basically what this is all about. And some of the activities that it is very learner-centered. The school clubs need to be run by the learners. So if you need to know how, please contact me and I will help you with that. And now for the cyber club, what are we going to be doing? It's online activities, hands-on activities, whether it's technology, whether it's, well, today you're going to be exposed to scientific investigations with Dr. Tanya, Reinhardt, and there's skills training. Of course, we're going to cover aspects about Expo project. There'll be competitions, inter-club competitions, right? And it's going to be various presenters from various fields, the East Stream fields. So we've got WESA, we've got maths activities that's going to be done. We've got super scientists. There's UniZulu Science Center. There's a whole host of organizations that are coming on board. Just a quick one about the dates for the Expo, right? You can contact me again if you're interested in the dates. And today we're going to focus on the scientific investigation type of project, where you're going to be conducting experiments, testing hypotheses. That's the focus of the scientific investigation, okay? And then there's these 13 categories. So while Dr. T is doing some of her experiments, today on DNA, you might want to think about which category. Is it medical science or which category? So these are the 13 categories in which you can do your project. And these are the 4 types of projects. As I said, today we're focusing on the scientific investigation, hypotheses, testing, where you're going to be using equipment, okay? We will get through to the other types of projects. And this is how you can get hold of us. You can get access to the resources. You can WhatsApp me, that's my WhatsApp number, our email, social media, just follow us on Facebook and so on for upcoming events, right? That's how we're going to share the online events, okay? So I'm done on my bit. I'm now going to hand you over to Dr. T to do the exciting stuff. Dr. T, over to you.
Okay, thank you, Nalini, and I hope it's going to be exciting. I'm not too sure, but we'll see. Okay, so I'm very excited because of this basically online club. The reason being is it's been in the pipeline for such a long time. And let me just show you. Okay, and I'll just need the floating menu control. It's been such a long time. Sorry, I'll just have to go back. You see, this is all the things when you start doing things. Okay, all right. And Nalini and I have been talking about it for years and years that we want to do an online science club, and now finally this is our very first session. As I said, I'm very excited. So today it's basically DNA, the Discover the Recipe for Life, and that's me, Tanya Reinhardt, also known as Dr. T, and I had help from Zimdela and Tembelani, my 2 interns here in the Science Center. Okay, so what do bats, sheep, fruit, flowers, E. coli, bacteria, fish, snakes, you and me have in common? All of these ones here basically have in common that they all contain DNA. So it's us that contains DNA. It's all these characters that you see here that contain DNA. So it's also known as, or DNA is also known as deoxyribonucleic acid. And this is basically the chemical name for the molecule that carries genetic instructions in all living things. So, and as you can see, this is just a minute, a tiny example of all the varieties that contain DNA. So and here we go, the first technical glitch. Okay, so where can we find DNA? Most DNA is located in the cell nucleus, so where it's called nuclear DNA. But a small amount of DNA can, for example, be also found in the mitochondria, where it's called mitochondrial DNA. Makes sense, eh? And DNA is basically in every cell. And chromosomes are found carrying your DNA in the nucleus of your cell. And here you can see a nice little sketch of the cells, the nucleus, the chromosomes, the DNA that's part of the chromosomes, and what a gene is, basically a segment of the DNA. So, what do you think? How many cells do you think we have as humans? And it's absolutely amazing. We have round about 37.2 trillion cells. And this is what the estimate that scientists have. So this is the number that the adult human body contains. So round about 37 trillion human cells. Okay, so now comes the exciting part, hopefully. And what we're going to go through is just quickly what you will need for the experiment, to do the experiment. I hope that you have everything ready that was on the list. So what we need is a banana, some salt, some hot water. Please do not use boiling water because we're putting it in plastic, a cup, some measuring spoons and a measuring a measuring cylinder, some plastic bags, some glass, and some filtering material. You can either use cloth or you can use other things like tissue and so on. Methylated spirit, and some sunlight dishwashing liquid. Okay, so hope you have everything ready. Okay, so as you can see, you can, as I said, you can either use like paper tissues, like serviettes or so on, but I actually prefer cloth, and I'll tell you in a minute why we actually would like paper cloths. So let's just start by extracting the DNA. So what you need to do is you need to peel the banana. Okay, you're going to chuck the banana into your Ziploc bag or plastic bag. And now you're going to start, so I close it, and now you're going to start mushing the banana. So squeeze it hard, and basically what this helps with is to expose as many cells as possible to the chemicals that we are going to add. Okay, so here you can see I'm squeezing, right? And this is luckily pre-recorded, so I can talk while I'm doing while I'm showing you this. And so we are spreading things out. So we are increasing what we call the surface area. Okay. So we're basically breaking apart all the cells. Okay, a little bit more, and we've got 5 seconds left. Okay, so this should do the trick. And you can also see here at the bottom, this is basically also some instructions where you can find how to extract banana DNA. So now in the next step, what we need is we need the hot water, and we're going to add half a cup of hot water into this. And what we're also going to do is, I'll just need to do this. We're going to add 1 tablespoon of salt, and you saw that I'm basically moving my finger across the measuring spoon. This is just to make sure that it's a consistent amount of salt. So basically a level amount of table salt. Now you're going to start stirring, and you just make sure that it dissolves quite nicely. In the next step, we are opening our plastic bag with the banana mush, and we are going to add the salt water. Okay, So the salt will basically help the DNA to stick together once it's out of the cell. So you can see I'm kind of mushing it. You can shake it a little bit as well. Just make sure that it doesn't foam too much. Just gentle process, a little bit of massaging, and what you're doing is you're just exposing the maximum of banana cells to the salt water. Okay, here we go. All right. I hope that everybody can follow so far. Just smooshing it. Okay. So we're doing it for about 45 seconds. Okay. In the next step, what we're adding is we're adding dishwashing liquid. Okay. So the dishwashing liquid, so we're going to squeeze it into half a teaspoon. Okay. Just nice and level. And we're going to gently mix the dishwashing liquid, and we're trying not to make it form too much. So basically what helps, the soap helps basically with breaking apart the cell membrane and the cell parts that hold the DNA. So if you think about it, dishwashing liquid basically removes the fatty outer layer with it, of it, and it attaches to it and it breaks open the cells, and this exposes the DNA. So please do not let the dishwashing liquid froth and foam too much in the mixture, as it may prevent the effectively extracting the DNA. You can see I carefully expose everything, mix everything up, and as you can see, there's very little foam in the plastic bag. There we go. Squash. Right. So now we are going to sift through. So what I'm doing here, and I'll just quickly stop the video. So just to explain, there are different ways of how to basically extract these kinds of things. Okay. So here I've got a sieve and my paper towel. I prefer basically to use a sieve and a paper towel, a sieve with a paper towel, because the paper towel sometimes does break. So remember when you have your paper tissues and paper towels, when it's getting wet, it's actually quite weak, and so sometimes it might crack. So as you can see on the side, there's also a cut off bottle that you can also use just as a support. Let's continue with it. So here we go. So this is the bottle top, just to cut off. So it just, as I said, just a support. So there's still space for the liquid to dribble through in the middle. So you would secure it there and pour it through the paper tissue. But my favorite is still the cloth. This is just a simple cleaning cloth. You can also use an old t-shirt or an old silk stocking. That's also going to work. And you see, I'm basically securing it just with a simple rubber band. And I'm basically making a little indent. So don't make it too taut, because otherwise it's going to be a big mess. So I make a little pocket inside. And now I'm basically opening the bag and I'm carefully placing the liquid inside. So, and bits as bits, and now you can see why I made this indent. So if I would have, as I said, if I would have put a taut, then the whole liquid would kind of spill onto the sides. There you go, a little bit more. And here you can see I'm just lifting it up, so you can see the clear, so the liquid basically going into the glass, which contains now the cells, or the broken up cells. Right, a little bit more. And I'll show you again how it's going to work with the paper. So you could also basically probably just put it through this fine sieve, but I like to place the paper towel in it. The only problem that I usually have with it is when I'm inserting it, that it takes a long time for it to filter through. So with the cloth, it's actually fairly quick and so on. So you can see, remember with the other one, it was just streaming through, but this one just dribbles through. Okay. And as you can see, it's a big mess on the side. I'm always a little bit messy. So if you have a table and you cover it with plastic, that would actually help a bit. So don't squeeze too hard, because then your paper towel might, break. Okay. And just also quickly going to stop this. So now what I'm adding is, and it's quite important to have ice cold methylated spirit. So you can usually put it in the fridge or in the freezer beforehand, before you're going to use it. Just remember, methylated spirit or alcohol is poisonous and flammable. So please do not drink the methylated spirit or work near an open flame. And also, I know that some kids often have difficulties with the methylated spirit, that it's very smelly and so on. So best is to work in a ventilated area. Okay, so what I'm going to do now is I'm going to basically now tilt the glass and pour the methylated spirit carefully in, so that I'm basically getting 2 layers. So you want the alcohol to stay on top of the banana mix. And this helps because the DNA doesn't dissolve in alcohol. So this helps the DNA to basically to form clumps of DNA. So here you can see I've got a big nice layer here. All right. And now what we just have to do is we now have to wait for about 8 minutes to do so. So while we're waiting, let's just quickly run through a couple of things. So, of course, you might be interested in how we could basically turn this into a research project. You could, for example, use 2 different approaches. You can use this method to extract DNA from different kind of fruit or vegetable, or you can use different methods to extract the DNA. And this is actually quite interesting. And this is also what scientists do. They're not only looking at what you can do, but also on methods, because methods, developing different kinds of methods brings actually scientific investigation forward. So what would be, for example, what can the research question or hypothesis be? So, for example, we could say, can we use this method to extract DNA from any kind of fruit and veg? And you also have to look at your constant. So this is quite important, that you basically, in this case, for example, you have the prescribed steps to extract the DNA. So stick exactly to the same time. Do this first, then you do this, you use the same chemicals and so on. But the only thing that is going to be a variable would be, for example, the different kinds of fruit and vegetables. Now, of course, you can look at, can we use actually the first step of mushing for every type of fruit or vegetables? And you can probably answer the question yourself. Think about potatoes or think about mushrooms. Can you really mush potatoes or raw potatoes? I would say not really. The question, of course, then is, how can we break them down? And I'm going to show you some methods how you can break things down. Also, what do we have to consider when we do the experiments? And as indicated earlier, the steps and timing need to be exactly the same. So you cannot, whenever you do a scientific experiment, you cannot put additional variables in by saying, oh, okay, so now I'm mushing for 45 seconds, and then the next step you're going to say, oh, I'm going to mush for 25 minutes or something like this. So it needs to be constant. Also, what do we have to, so with the steps and the timing basically need to be exactly the same. So let's look at the different methods to extract DNA. So what could the research question there be? So we could basically look at which method yields the, and I put it in inverted commas, the most DNA. What would be your constant? So the type and size of fruit. So for example, you use half a banana, but just keep in mind that, bananas come in different sizes and shapes, and half a banana might not be the same as half the banana. So for example, you could rather use a fixed kind of weight. What would be your variable? So for example, the various methods of modifying the method currently used. So for example, you could investigate if, by adding more or less salt, it has an influence on the DNA. Does it have an influence if I change the temperature of the methylated spirit? Does it have an influence how hot the water is that we are going to add to it? So all these ones are the kinds of things that you can vary. Okay. Also, how can we measure which is the better method? So, and here you can use, for example, a quantitative and a qualitative approach. Qualitative means observing. So, for example, you could take a photo after a specific amount of time and compare the amount of time just in the photos. You can also try and do it sorry, that was qualitative. So you can also try to do it quantitative. So you can try and filter the DNA and, to maybe weigh the DNA that you extracted. But, by this method of comparing things could be, for example, a research project in itself. The other thing is, what do we have to consider when we do the experiments? Remember to keep the exact amount of banana. By varying the banana, of course, then you're changing basically the amount of cells that you have. So as I said, it's also interesting to find one way to compare the amount of DNA and then stick to this method to compare the DNA. Okay, so these ones are just some ideas, as I said, some ideas that you can use to do this. Oops, and I'll just have to, okay, it's not advancing, here we go. Here again, we have some examples of how to prepare the samples. So with fruit like banana, fruit like kiwis, fruit like strawberries, it is actually easy. So you could use the same method that we've done here. You just kind of mush the fruits, the veggies, the fruits, and then this would be. For vegetables, so you could, for example, put them in the blender with some water and salt at the start. If you want to see how grains behave, so you can soak the grains in hot water to a specific amount. So, for example, 3 to 10 minutes at the start, and then process it from there. For beef liver, you can soak the pieces in fresh pineapple juice or contact lens solutions for a day first. So this would break up the proteins that protect the DNA in the cell. Or for your cheek, you can actually extract your own DNA. You swirl a mouthful of salt water around while you shake your cheeks. So this sounds not so nice, but anyway. Okay, so just to emphasize a little bit on the background, and this is also something that when you do a research project, that's quite important, that you do a background research. So the internet is full of information, but it's also sometimes good to read what scientists call a scientific paper. So in a scientific journal, if you have a good understanding, or you're able to understand what they're saying. So libraries are also good places to go. Books are also good places to look up. So what is the purpose of DNA? So in every living creature on Earth, everybody has DNA. This is what we established right in the beginning. So, and this is basically the special set of instructions that makes us who we are. So this determines, if you have blue eyes, brown eyes, if you have curly hair, straight hair, blonde hair. White tigers have stripes, but leopards have spots. it all boils down to the DNA, and it's really quite exciting. And there are huge projects from scientists how to decode the DNA and look at which section of the DNA is responsible for what. Okay, for those who have never ever seen this, how does a DNA structure looks like? Actually, think about it as a ladder. And think about it that you twist this ladder. So think about it as a rubber ladder that you're going to twist. And this is also called a double helix. And the sides of the ladder are made out of sugar and phosphates. And the rungs of the ladder are made out of 4 different chemicals called bases. And the name of the bases are adenine, thymine, guanine, and cytosine. And just like a jigsaw puzzle, only specific bases match with each other. So you have your thymine that goes with your adenine, your guanine that goes with your cytosine. So this is always the case, okay? So if you have, for example, only one rung of the ladder, you are able to complete the other side because you know exactly which base pairs will match, will go with the other one. So if you have, for example, a C on the one ladder, on the one rung of the ladder, you know exactly that on the other side there needs to be a G.
With all the billions of people in the world, it's amazing to think that we're all different. When we're born, each of us grows according to a unique set of instructions that are...
Sorry, guys, I just need to stop the floating control.
...inside every cell in our body. All living things are made of cells. They're like small packets of life. Inside each cell is a nucleus where the instructions are stored. This instruction manual is called DNA and contains all the information needed to make you, you. With billions of letters making up each person's manual of instructions, our DNA is huge and is coiled up to fit inside every cell in our body. But how does DNA actually work? On its own, it can't have an effect because it's just the instructions. Cells are like tiny factories that follow instructions in the DNA to create proteins, the building blocks of every part of our body. To create a protein, first the instructions have to be identified in the DNA at the nucleus, almost like a highlighter pen on typewritten text. Then, this instruction is copied, in other words, transcribed, into a message called messenger RNA that leaves the nucleus. This message is then translated to the language required to make the protein. It's rather like IKEA. In the factory are the golden instructions to enable all the different furniture to be assembled. This is the DNA. If you're getting a table, then just those instructions are copied and inserted in the packaging with the panels and screws. This is the messenger RNA. Then you must assemble the table according to those instructions, like a factory making the proteins. Proteins don't last long, typically a couple of days or even less, so they need to be constantly produced to keep your body healthy. Proteins have many jobs in your cells. They carry the information to make your brain, stomach, muscle, all of you. 3 important functions of proteins are building tissues and muscles. Protein helps to make you stronger. Digesting your food to help you produce energy. And strengthening the immune system to fight off infections. So your DNA is there inside you. It's working all through your life to keep you healthy and making you become the special individual that you are, along with everyone else and every other living thing on our world. In many ways, we're all very similar. 2 arms, 2 legs, hair, and so on. And yet, we're all different. Even in the largest crowd, we can recognise our family and friends just by how they look. When you think of the population of the world, the potential for variation is huge. And it's all achieved with DNA.
Okay, for those who don't know, IKEA is a European furniture manufacturer. So you can think about it just like as our surprise home, where you can buy furniture that you can assemble. And this is basically how you can see your body as well as some furniture that people assemble. Okay, so now I'm pretty sure that we've got some time, and if you've done the experiment, this is basically what you should be able to see. This gooey stuff, the snot that comes out of it, that you can see floating now in the methylated spirit, so in the blue part. This is your DNA. So this is what you're basically seeing. So this snot here, that is your DNA. And I've just got some stuff here. Let me just show you. And where is it? Here we go. So I'll just hold it up. I'm hoping that you can see this. All the stuff here. This is basically DNA that we've extracted from the banana. Okay. So just a little bit of a recap. So what we've done, so we opened the plastic bag, we added half a banana into it, we mushed it for some time, we added 1 teaspoon of salt to half a cup of hot water. We added the half of cup of hot water into the mix. We gently mixed everything to break apart the cell membranes. So we let it stand for a short while. So while mixing it, we added dishwashing liquid to the whole mix. We mixed that one again just to break open the DNA or remove the outer layer of the cells. We placed some cloth into secured a cloth around the glass. We poured the mixture in it. We filtered basically the cell DNA mixture, saltwater mixture, and put it into a glass. And then we added some methylated spirit by tilting the glass and added a layer of about 2.5 to 5 centimeters. And then we let it stand for about 8 minutes. So as you can see, it is a very simple process that you can do with very simple material, and you can extract, this kind of DNA. So just to finish off, and I'll just try to do this, just to finish off, just some careers that are related to DNA, if you want to know, what people actually do with it. So microbiologists is, for example, they use DNA to find out the DNA of small organisms and see how they function. Geneticists, of course, they want to find out, genetic relations, for example, between parents and children, also look at genetic diseases and lots of other different things. Biochemists also look at these kinds of things. Crop scientists, of course, they want to create, the most juiciest fruit or, drought-resistant plants, and they often use also DNA experiments or DNA to basically extract the right genes that they would need for their plants. Anthropologists basically look at, the DNAs and how human evolved and who's related to whom and so on. Medical scientists now, of course, with COVID and other medical issues like HIV and so on, DNA is quite important. Crime scene investigation, if you look CSI, you know that forensic scientists also use DNA to find the bad guys or who were at the scene and so on. And even biologists use DNA as well to see relationships. So for example, you can basically determine from where an elephant, from which area an elephant is coming from by just looking at the DNA. And yeah, this was it from my side. If you have any questions. You can unmute yourself if you have any questions. Okay, I can see in the chat. Okay. We address the lack of resources and activity-based learning opportunities which have resulted in poor understanding of key science. We have science club in school across the country registered with us. Okay, all right. Okay, Lungile, thank you. Any questions? No questions. Okay.
Tanya?
Right. Yes.
Not a question, but just to give some ideas to, because I see there's lots of teachers that are here. So what can happen is the session, for example, is recorded. Now the learners that are coordinating the science club, they can watch the video again, they can learn how to do the extraction process, and then they can invite other learners to come and watch the experiment being done. So that's something, you know, on this platform of the Cyber eStream Club that we want to do. We want to provide an opportunity for the science club itself to find activities to do, you see?
Yeah,
so even if learners,
so what we are just doing is we're just giving some examples of what you can do. And often what you don't really need is you don't really need expensive equipment. You can of course get the expensive equipment, but we're trying to basically use simple material that everybody has access to some extent. Yeah. Also, if you you can also contact us if you have a project that you want to discuss and where you want to get some ideas or want to bounce off some ideas. You can also more than welcome to contact us here at the university or via Nalini.
Yeah, thank you so much for that, Tanya. And I really liked the amount of project ideas that you've actually shared. So just from this activity alone, if there are learners that are looking for projects within this field, you've given us so many examples, and that for me, that was the takeaway that was really great. So thank you. I don't know if any of the teachers want to say something or ask any questions. Any teachers or learners? I know there are some learners that are one of the schools, Grantham Park has some learners that are here. Anyone?
Yes, can I say something?
Yes. Yes, go ahead.
My name is Dookie. I actually saw this on Twitter today, and I was like, I didn't know what I was getting myself into, but I was very much interested in seeing what it has and what it's doing, because I did say, I have a connection with teachers and science clubs in schools, especially in rural areas. So I'm always looking on the lookout for activities that they can do on their own, just to get engaged and to learn more and also have fun doing science. So I'm happy that I ran into this and I was able to attend as well. Thank you for having me. And I don't have a question, it's just a comment to say thank you, and I've learned so much. And I don't know, does this happen, how often does this happen? Because I would love for maybe one or two of the teachers that I'm in contact with to be able to register and take part as well.
Nalini, you want to answer this?
Okay, thank you so much, Lungile. I see you from Science Plaza, which is fantastic.
Yeah.
So as Tanya mentioned in the beginning, this is something that we're piloting in KZN, and this is going to be an ongoing session. So we're looking at alternate weeks, so 14th, and then we skip a week, and then we're going to have another session at the end of February. So it's going to be every 2 weeks. We also wanted to
Okay.
give schools a break to do other activities, besides just only watch. So those schools that already have their science
Okay.
clubs, there might be other things that they want to do. So we didn't want to just,
Yeah.
restrict them to only attending our online sessions. So we will be
Okay.
having ongoing, and the idea is for the school science clubs to register with us so that we have their
Mm.
contact details and we can send the information. So whenever we're having a session, we send the
Yes.
info to those schools.
Oh, great. Thank you. Thank
Okay.
you so much, Nalini.
Yeah.
Thanks.
Okay, I can see that Sajida Gora has got a question. You wanna ask the question?
Yeah,
Hi,
sure. Go ahead.
hi, I'm from Grantham Park. I have a student here that has a question for you.
Okay, awesome.
Hi, my name is Dandun Zele from Grantham Park Primary School. My question is.
Hello, how are you? What
Go
is
ahead.
your
I'll
question?
talk at the mic.
What is your question?
My question is, how large is a banana?
Don't be shy.
Can you hear us?
Not yet.
I can't hear her. I can hear you, but I didn't get the question that she was asking.
Right.
My question was, how and why does a banana have a DNA if it's not a living thing?
A banana is a living thing. Not really living things, but all the things like fruit and all the other things, they do actually contain DNA. So also all the fruits that you have, they all contain DNA. Okay.
So Tanya, can we say that things that were once living? Because now the
Yeah,
banana is.
we could probably say I'm not a biologist, but I'm pretty
Mhm.
sure yes we could say things. So for example, take your bacteria for example. They also contain DNA. Interesting enough.
Mhm.
So it was once a kind of plants are kind of living organisms and so on, and this is just the fruit of your plant. So it's part of your plant. I wouldn't say that it's there.
Mhm. Go ahead.
But since once it's broken off a tree, is it still a living thing?
This is more, I would say more like a philosophical question. Is it still a living thing? I actually don't know. I have to say, I don't know. And it's a very good question, in fact. And this is the kind of question that maybe you want to research and look. So you could, for example, turn it into a research project and say, how long actually can you extract DNA from things? So remember, you can even extract DNAs from bodies that are dead. Remember? Okay, so when forensic scientists investigate things like murder or things that happened a long time ago, they can also extract DNA and determine the person's identity using the DNA. So I'm not too sure when actually DNA kind of, and I put it in inverted commas, dies. But apparently, so from what I can see is that the genetic information is stored in the DNA for quite some time. Does that answer your? Does it answer that question? So you saw that I was navigating around it a bit, hey?
Yeah, thanks,
Yes,
Tanya.
I kind of thank you for answering my question.
Okay, pleasure.
Yeah, but
Great
the
question.
banana.
Great question.
Yes, ma'am.
Sorry, I said the banana, if it rots, it doesn't really die, isn't it? Turn into microorganisms eventually and become nutrients for the soil. So there's no real death in it, isn't it?
Yeah. But it would be quite interesting to see how this whole process is. As I said, I'm not a biologist, so I won't be able to talk about this. I can talk to you about dead rocks and stuff like this. So yeah, maybe you want to investigate it. So maybe you could do your own research project and say, oh, okay, I'm looking. I want to see if the DNA gets less and less over time. So you take the same banana, you cut it into equal size, pieces, and then you say, oh, okay, so I'm gonna extract DNA from a slice and see how much I get out from after 2 weeks, 3 weeks. A month, 2 months, which is then going to be really growth, and so on. Maybe that's an idea.
Thank you, Tanya. I actually have 20 students here and they're loving your experiment, actually.
Oh.
And it's the first one for the year. So thanks a lot for your assistance.
Thank you. There's a hand raised.
Monica Shazi has got a question. She has got her hands up.
Hello.
Hi, how are you?
I'm fine, thanks, and you?
I'm very well, thank you.
Yes, firstly, I just want to say thank you for a brilliant presentation that you have done on DNA extraction. I had no idea, but now I have an idea. Thank you so much.
Pleasure. That's great.
And then two, I wanted to know, it's my first time to take part on this. So I was wondering if it's going to be an ongoing thing. And then
Yes.
if one wants to register full time, how do you go about registering?
I think Nalini can help you with this. Yes, it's going to be an ongoing thing, not necessarily with me because I'm, there's going to be some other brilliant organizations like the UniZulu Science Center and I think VESA is going to come on board. That also, so just to expose you to different kinds of things. So you're gonna see me in a fortnight as well. I still have to think about what I'm going to do with you guys. So please join us again and just spread the words because I think it's quite important to expose the learners to as many different ideas and things and to show them that science is not necessarily boring, but can be really exciting and asking questions and being curious is really, really brilliant.
Okay. Thank
Nalini,
you.
you want to say a little bit about the club?
Yeah, so Tanya's quite right. We're going to be repeating these online sessions fortnightly. And like I said, it's to create a network also.
Okay.
Network of school clubs to support expo activities in your club. Like right now, we're focusing on project ideas. And as you heard from today's discussion, there's so many project ideas that have come up from just, and it's about learners asking these questions and then trying to find the answers themselves. So I love that conversation with, with the learner from Grantham Park, because now that is something she can go and investigate as a young scientist. It's stuff that they're not learning. Yes, they're doing the section of living and non-living in science in the curriculum. But now this learner can now take it a little bit further than just learning the content, you see? So that's the idea of these sessions, not only to, help you with your expo projects, but to make learners see that science is real and they can do it. They can do these experiments. Right.
Thank you so much.
Justice Nkela is got a question as well. Yes.
Not necessarily a question. Thank you so much, ma'am. Thank you so much for the wonderful presentation that you've just shared with us. Though I could not join exactly at 2:30 due to network challenges. I wanted to check if it's possible. I think it was recorded. If it's possible for this recording to be shared with us so that we can go over it again. That's the only question or concern that I may be having. Thank you.
Okay, sure.
Yeah, Nalini, you recorded it and you're going to place it online. Is that correct, Nalini?
Yeah, we will. But you can. Is my contact details coming up there? You can just send me a message or WhatsApp or email to say that I should send you the recording. I do realize there's, the notice was short and some schools didn't get a chance of collecting the materials beforehand. So yes, we can provide you with the recording. And we'll share it on our YouTube channel as well. Okay.
Thank you so much, Nalini. Thanks a lot.
Okay.
Right. Are there any other questions? Okay, it doesn't look like it. So I'm basically done on my side. Thank you so much for coming, attending, and I hope to see lots of you back in 2 weeks' time. As I said, I haven't decided on what I'm going to do. I'm still looking into things, yeah, I hope it's going to be as exciting as it was today for me. Okay, thank you so much.
Thanks for your time.
Thank you,
Thank you.
everyone. Hoping to see you
Thank
again
you.
in 2 weeks.
Thank you very much.
Okay.
Take care. Have a good week.
Thanks.
Thank you very much,
Bye,
Nalini.
everyone.
Thank you very much, Dr. Chi.
Okay, pleasure. Okay, bye-bye, guys, and have a lovely rest of the week.
Very
Okay.
much.
Bye-bye. Bye.
Thank you.
Bye,
Bye.
Bye, everyone.
Bye.